So hi, good afternoon, everyone, and thank you for joining us for our Canals Brown Bag presentation today. I'll start by introducing our first presenter of the afternoon. Katie Lohr recently completed a PhD in Fisheries and Aquatic Sciences at the University of Florida. Her studies focused on strategies for increasing the efficiency and effectiveness of Acroporid coral restoration. Prior to completing her PhD, Katie spent several years working on coral reef conservation and restoration projects at the Central Caribbean Marine Institute in Little Cayman, Cayman Islands. She previously earned an MPS in marine biology and fisheries at the University of Miami and a BS in biology from the University of Michigan. Katie, take it away.
All right, thanks so much, Christine, and thanks so much to everyone for being here and listening online. Today I'm going to talk about two chapters from my PhD dissertation that are focused on ways we can incorporate genetic information to support ongoing coral restoration. Okay, so there we go. Okay, so we'll first start by talking about the Acroporid corals. So Acropora is actually the most speciose genus of coral in the world. There's about 180 species globally, but in the Caribbean, where I've worked throughout my career, we only have 2 of these species. So the top will be the focus of my talk today, staghorn coral, Acropora cervicornis, but we also have the elkhorn coral, Acropora palmata. And although we only have these 2 species, they have really important ecological functions on Caribbean reefs that we'll get into. I do want to note that technical difficulties. Okay, like all reef building corals, these species have a symbiotic relationship with a dinoflagellate that lives in their tissues, and this dinoflagellate provides them with 90% of their nutrition through photosynthesis. And when the symbiosis breaks down, that's what's known as coral bleaching. So that will come up again. So just a little reminder about how that works. Historically, both of these corals were spatial dominants. So they would form these really vast monospecific thickets that created really important habitat for other species. They also have a fast growth rate. So each one of those branches can grow about 10 to 20 centimeters per year, which is really fast compared to some of the mounding species that might grow maybe 1 centimeter in diameter per year. That means they're really important for maintaining positive rates of reef calcium carbonate accretion, so keeping that reef structure. They are also complex. They have this sort of rough branching structure. As I mentioned, that creates habitat, but it also adds roughness to the reef, which is an important factor in reducing wave energy and protecting shorelines. So this is what I mean when I say spatial dominance, right? So you can sort of see nothing but elkhorn coral as far as the eye can see in this photo from Florida. And same for staghorn coral, this example from Jamaica. But unfortunately, over the past several decades, both species have declined up to 95%, in some areas more, throughout their range. That decline has been largely driven by disease. The causative agent of that disease is still a hot topic of study, but these corals have also been affected by warming that creates coral bleaching. Prolonged temperatures can result in mortality. Also, other factors like declines in water quality and loss of herbivory have also impacted these species. So in response to this sharp decline and the ongoing threats that these species face, both of these corals were ESA listed in 2006 as threatened. And when a species is listed under the Endangered Species Act, a recovery plan is drafted. The recovery plan for these 2 species notes that we have to reduce or eliminate threats to recover both species. That includes local threats like poor water quality, but also global threats like climate change. It's also important to generate some ecosystem level improvements, so recovering processes like herbivory that have been lost. And then finally, the recovery plan notes that active restoration is going to be a key strategy to recover both species. When we're talking about active coral restoration in the Caribbean, that is most often going to be referring to coral gardening, and that's going to be what this talk focuses on. Coral gardening refers to this process where you can take a portion of coral skeleton and tissue from a healthy wild donor colony. that actually mimics natural asexual fragmentation processes that these staghorn corals will use in the wild to reproduce. The fragments that are collected can be established in ocean or land-based nurseries. This is an ocean-based example you see here. When these corals are in a nursery, they are kept away from competitors like algae, corallivores like snails, and they can grow quite large with that fast growth rate relatively quickly. At that point, they can be fragmented again to create a self-sustaining stock of coral in a nursery. And because these are being propagated through asexual fragmentation, essentially, all corals propagated from your initial sample are going to be cloned. So you can also maintain distinct sets that come from the same coral genotype. The ultimate goal of this process is to outplant these corals back to the reef where they can immediately add structure and habitat and ultimately grow to maturity and contribute to species recovery through natural reproduction processes. Unfortunately, the recovery plan notes that because of sort of the magnitude of threats coral reefs are facing and the few individuals we have left from these species, right now it's going to take approximately 400 years to achieve recovery, and the total cost of recovery actions is upwards of $250 million. So there's a lot of interest in finding ways that we can improve the efficiency of restoration and help provide these restored corals with a way to survive under future ocean conditions. So that's where this sort of idea of using phenotypic information and integrating that into coral restoration processes comes in. So what kinds of phenotypes are we interested in? Disease resistance is one good example. So on the left, you see a healthy coral colony that's in contact with the diseased colony. Through the contact, that colony has become diseased and unfortunately succumbed to that disease. On the right is the same process, but with a disease-resistant coral. And you see that that coral has been able to resist infection and survive. So traits like disease resistance, if we could identify genotypes that have those traits in nurseries, and then integrate that into current restoration strategies, that could potentially improve the overall success of restoration efforts. So understanding that we need this information to support restoration, we conducted a study looking at a variety of traits in a nursery to see what sort of variation we could determine. We already know that growth rate can vary among genotypes. That's something that's been well studied in nurseries in terms of total linear extension, so how long each branch is growing. But even so, phenotype is rarely evaluated systematically in nurseries, and usually a variety of phenotypes aren't considered. So we conducted our study in an established Coral Restoration Foundation nursery in Tavernier, which is in the Florida Keys National Marine Sanctuary. And we used 10 known genotypes that had been previously identified through microsatellite genotyping at the BALMS lab. And so for each genotype, we collected 4 5-centimeter non-branching apical tips, which is essentially a coral branch tip that looks like that, from each of 3 broodstock colonies. So that gave us 12 tips per genotype. Labeled those and haphazardly distributed them across these PVC tree nurseries. That's a really common grow out method that's used for this species in the Caribbean. And then we observed and measured these fragments over a little more than a year and collected data on total linear extension, so linear growth of the corals. Also looked at colony condition, so looking for signs of bleaching, for example. And then we also removed the corals from the nursery at 3 time points and weighed them. We used buoyant weight, which gives a good measure of calcification. So how much calcium carbonate is actually being laid down into the skeleton as the coral grows. All right, so here are some of our results. I'll first have you guys look at panel A. On the x-axis, we have time in days. The left y-axis is total linear extension. And then the right y-axis is temperature, which is shown in the dotted line. So by the end of the experiment, ANOVA indicated that there were significant differences in total linear extension among our 10 genotypes, and the letters denote post hoc differences. We did observe bleaching about halfway through the study. That's indicated by the arrow. And a paired t-test indicated that growth overall for all of our genotypes decreased as a result of the bleaching incident. For buoyant weight, we found the same result, significant differences in our 10 genotypes that is shown in panel B. This is the same data shown in a little bit of a different way. So this time on the x-axis are our 10 genotypes. The left y-axis shows total linear extension, which is the black bars. The right y-axis shows buoyant weight, which is in gray. So what we tested here was whether there were differences among our genotypes in the ratio of buoyant weight to total linear extension, and we found that there were, and that's what those letters denote. We also found that as total linear extension increases, the ratio of buoyant weight to total linear extension decreases. Or in other words, our fastest growing genotype U41 has less calcium carbonate per unit extension compared to our slowest growing genotype U25. And that could have likely implications for skeletal density. So even though U41 is growing faster, its skeleton is likely not going to be as dense. As I mentioned, we observed bleaching and, okay, we're back. So we also found significant differences in bleaching prevalence among our genotypes. This is just a visual representation of some of those differences. Each of the rows in this grid shows a different genotype. And U25 had the highest bleaching prevalence of any genotype. Okay, so ultimately, we found that there are a variety of traits that are relevant to staghorn coral restoration that can be discerned in a nursery. The big question that still needs to be answered is whether those phenotypes are consistent once the coral is outplanted. So there's been a lot of evidence to suggest that total linear extension in particular is a pretty plastic trait, so highly dependent on what reef the coral is planted to. So that's still a big question that needs answering. As I mentioned, we found a trade-off in extension versus buoyant weight. This is consistent with studies from other nurseries, as is the diminished growth with bleaching. One limitation of this study is that we weren't able to genotype the symbionts that were associated with each of the 10 genotypes in our study. Most staghorn coral is inhabited by the same species of Symbiodinium, but whether there was an effective symbiont, we still need to look at that. However, both symbiont and host have been shown to play key roles in bleaching prevalence. The other thing I want to note is that this process is fairly time and labor intensive. So coral restoration practitioners are quite focused on, producing corals and outplanting those corals. That's a lot of work, a lot of time and resources. So some of these practitioners, most of these practitioners probably don't have 390 days to go out and be measuring all of these traits repeatedly over time. So that sort of brought us to our next question. Because these in situ measurements can be impractical, we wanted to find if there was a more streamlined method to be able to discern key phenotypes among our genotypes of interest. And so for that, we turned to metabolomics because phenotype is linked to the metabolome, and I'll walk you through exactly how that works. Okay, so most people are familiar with the concept of the genome. It's the full set of DNA within an organism. That DNA is transcribed into RNA. The full set of RNA is the transcriptome, and then RNA is translated into proteins, and we have the proteome. So those proteins catalyze a number of biochemical reactions within an organism, and the small chemical compounds that are the reactants, intermediates, and end products of those chemical reactions are known as metabolites. So things like sugars and fatty acids, those are primary metabolites that are involved in regular growth and reproduction processes. We also have smaller chemical compounds, secondary metabolites that aren't maybe necessary for regular growth and reproduction, but have ecological functions. The full set of those primary and secondary metabolites is known as the metabolome. Because the makeup of the metabolome is dependent on these sort of upstream processes of transcription and translation, there's a big genetic effect on the metabolome and what it's comprised of, but also because the environment can have an effect on protein activity, for example. The metabolome is also altered by the environment. So if we look at the metabolome, that really gives us a sort of good idea of what kind of physiological processes are going on in this organism at a given point in time. And because these physiological processes are really what underlie the phenotypes and functions that we see in the organism, there is a close link between metabolome and phenotype. And so that link has sort of been exploited in other fields. In agriculture, for example, there's a study where scientists looked at a set of different cultivars of potato, and they were able to find a metabolite biomarker that could reliably predict which genotypes of this potato would produce the best quality potato chips. So that was a trait of interest for that industry. But for our industry, coral restoration, we wanted to see if this sort of process could be applied to find metabolite biomarkers for traits of interest in corals. However, metabolomic profiling and studies of the metabolome, it's pretty new for corals. Previous studies have found unique metabolomic profiles among different species of coral. And metabolomic changes with altered abiotic and biotic conditions. But the question we needed to answer to sort of start down this line of inquiry was whether there are differences in the metabolomes of unique genotypes of the same species. So that is what we aim to look at. To do that, we selected 3 genotypes from that previous study. U41, our rapid grower with moderate bleaching prevalence, U44, moderate growth, low bleaching prevalence, and then U25, which was kind of our dud with slow growth and high bleaching prevalence. So we sampled 5 to 6 replicate tips per genotype, and those were taken from 2 broodstock colonies and processed at the Southeast Center for Integrated Metabolomics. We applied 2 types of metabolomic profiling. The first is proton nuclear magnetic resonance spectroscopy, or HNMR. The second was liquid chromatography mass spectrometry, or LCMS. Both of those methods sort of have different strengths and weaknesses. HNMR is really good for identifying unknown compounds and primary metabolites, but LCMS is a lot more sensitive, so it can resolve some of those smaller chemical compounds that might be more rare. But for the purposes of today's talk for time, I'm going to just focus on the LCMS results. And what we're seeing here is partial least squares discriminant analysis. So sort of like a PCA, it's a dimension reduction method, but this is a supervised method, so it takes into account which samples are coming from which genotypes, and essentially showing us which factors maximize those differences. So we are seeing fairly clear separation between our 3 genotypes, so that does suggest there are metabolomic differences. We also do see this sort of big spread for genotype U41 in the green across both components 1 and 2. That suggests high within genotype variability in the metabolome for that particular genotype. When we look at what's driving those differences, I know this is a lot to take in, but I will walk you through it. So the sort of names and numbers on the right, those are different metabolites. So each row in this heat map is a different metabolite. Some of them are just numbers because they couldn't be identified. Across the top, the blue, red, and green bars show our 3 different genotypes. And then the color ramp from red to blue shows relative abundance of each of these metabolites. So we're going to focus on one of these, catechin, that's highlighted in yellow. So you can see catechin was relatively more abundant for genotype U25 compared to our other 2 genotypes. Okay. So as I mentioned, we were able to resolve distinct metabolomic profiles for these phenotypically distinct coral genotypes. So that does suggest there is promise for future studies to find key metabolite biomarkers that could be linked to these traits. As I mentioned, we did have higher variability for genotype U41. One explanation for that might be intracolonial variation in genotype. So this has been found to be common in Indo-Pacific acroporids. One colony can actually have more than one genotype. Another explanation would be differences in epigenetic effects from our 2 different donor colonies, but a lot sort of more questions than answers, unfortunately, that I have at the moment for that. I do want to go back to that one compound we highlighted, catechin. Catechin is a flavonoid with antioxidant properties. It's been really well studied from a human health perspective because it's found in high abundance in things like green tea. So people are really concerned with potential health benefits for humans. But interestingly enough, there was a 2018 study looking at the mustard hill coral, Porides asteroides, that you see on the bottom. And in this study, they actually set up these Porides colonies in tanks and then increased the temperature. So they thermally stressed all of these corals And for half of them, they dosed the tanks with catechin. So they added catechin, and that was able to stop those corals from bleaching, whereas the control colonies bleached. So that suggests there might be a thermotolerance function. So very, very interesting area for future study, but unfortunately wasn't able to delve into that during my PhD. All right, so just to wrap it up, we found key staghorn coral traits that could be discerned among genotypes in a common garden setting. Those phenotypically unique staghorn corals did have distinct metabolomic profiles. Ultimately, we need to understand whether those phenotypes are actually predictive of what this coral is going to do in a restoration setting, because it won't be great to, have all this information about what these corals do in a nursery if that's not consistent once they're on the reef. And then more metabolomic studies. As I mentioned, it's quite new to corals. We do have a lab mate, Joe Henry, that is continuing these studies at UF, and one thing he's going to look at is whether these differences are consistent for these same 3 genotypes from year to year. So that's going to be a really important question to answer. And then the implications, I just want to briefly talk about. So I want to stress that practitioners should continue to maximize genetic diversity in restoration, even if we think this coral is a super coral in a nursery, right? It grows really fast, it looks great. There might be trade-offs present. We noted a trade-off between linear extension and calcification. However, others have suggested that there could be a trade-off, for example, in linear extension and disease resistance, or things that might have implications for the corals to survive under future ocean conditions. So for now, keep the genetic diversity high. And then phenotypic information can support new conservation and restoration methods. So one study I got to be involved in was looking at sort of the other route to restoration, right? So using eggs and sperm, so using sort of harnessing the power of sexual reproduction in corals instead of asexual propagation. Recently, advances have been made where we can cryopreserve those sperm and then be able to use those from year to year. These corals only spawn once a year, so cryopreservation really sort of allows us to conduct studies later on. And one of those studies looked at assisted gene flow. This was actually done in elkhorn coral, but for the first time, this study indicated that assisted gene flow is possible for genetically distinct groups of corals. And what that means is that we could introduce potentially adaptive alleles from somewhere like Curacao to a location like Florida. We actually used cryopreserved sperm that was collected in Florida in 2016, Puerto Rico in 2008 to fertilize eggs collected in 2018 in Curacao, and that was successful for all the treatments. So that would be a really exciting area to start to understand what traits are present and what traits can be used in studies like this. With that, I'd like to thank my advisor, co-authors, field support, and Coral Restoration Foundation for working with us to provide those corals. And thank you very much.
Okay, are there any questions in the room? Okay, and we do, oh, okay.
Thanks, that was a great presentation. Quick question of clarification about the pretty dire 400-year projection for recovery, and was just curious if that assumes no change in environmental conditions or factors in water quality, temperature, climate change.
To be honest, I do not recall, but I believe it's factoring in sort of the current projection that we're on, so sort of a business as usual scenario for climate change and those sort of factors. But yeah.
Thanks.
Any other questions in the library? Okay. Thanks. Great presentation, Katie. I had a question about a quick question about your methods. So did you take repeat fragments for your metabolomics? Was it from like a connected colony, or was it from separate fragments that were the same genotype? Does that make sense?
I think so. Let me go back to that.
So it's like, was it like separate?
both.
Like
So
separate fragments on that tree, or were they all from like that one fragment?
no,
for
so
that
we
genotype?
we took 2 to 3 branch tips from each of 2 colonies in this case. So we were limited in the amount of samples.
Yeah.
But so we used 2 broodstock colonies. So that's where we think that variation in U41 likely came from.
So for that one you used 2?
For each of the 3 genotypes, we
Okay.
used 2 colonies of that genotype.
So were the repeat samples from the same connected colony, or were those more similar than the separate?
That wasn't actually
colonies?
analyzed, but okay, let me go to the heat map. So that wasn't statistically analyzed, but if we look at the heat map, you can see for U41, I don't have this labeled, unfortunately, but it's cut off, but it is labeled by sample. And for U41, there was no consistency between the 2 colonies of origin, actually.
Okay,
Yeah.
that's interesting. It's just so
I
thought it would be more similar.
That would sort of explain more. So that also does point a little more toward possibly more than one genotype in the colony, because even samples from the same colony were showing differences. Does that make sense? Yeah.
Okay.
Yeah.
That makes sense. Cool.
Okay, so we do have a few online questions.
Okay.
All right. Actually, more than a few. There's a lot of interest. So this is similar to another question, but someone wanted to know, how are the restoration valuations developed? Our team has a number of projects also to incorporate valuation research.
Like valuation of restoration projects.
Mm-hmm.
So I haven't been particularly involved in, ecosystem services sort of aspects of restoration or evaluation studies, but yeah, I'm sorry. I can't speak to that.
No, that's okay. And then, someone also wanted to know, have coral nurseries even started keeping track of their genotypes? In the past, genotypes were propagated in a very haphazard way.
Yes, that is something that has changed fairly significantly. So genotypes are, at least in the larger coral nurseries that I have worked in, and even many of the smaller ones, are now, as standard practice, genotyping every coral that comes into the nursery and then keeping track of those through tags, typically, to make sure that genotypes are all accounted for.
And so, let's see. Oh, and then I think we just have a few people saying, fantastic talk, awesome talk, great talk.
Thank you.
Pass that along.
I appreciate that.
Yeah, okay. Anyone else here have a question they would like to ask before we end the session? Okay, so thank you, Katie, so much. We're going to take a quick break and be back with Larissa.